Do you want to learn about new innovations within the life sciences industry, specifically about new innovations within gene editing? Well, if so, stay tuned because today we're joined by two wonderful guests. Omar and Jonathan are researchers at MIT who are at the cutting edge of gene editing technologies, recently pioneering the new PACE gene editing technique. If you want to learn about how this can be used to potentially tackle diseases such as cystic fibrosis and Huntington's disease, please stay tuned. And before we dive in, if you're watching this on any of the online platforms, please do like, share, and subscribe, especially to YouTube, to make sure that more people like yourself can reach and find out this material and learn new things about the life sciences industry. I look forward to speaking to all of you very soon. Welcome, everyone. This is another episode of the Life Sciences Trends podcast. I'm very excited for this episode. I have the lab coat on today because we're going to dive slightly more into the scientific side of things, but of course, keeping things simple. We have some wonderful guests today, and they're going to be describing and explaining more about gene editing technology, the newest and emerging trends within the space. And before I progress and get a bit too excited, can you please introduce yourselves and explain a bit more about your background and experience?
Yeah, sure. So I'm Omar, and of course that's Jonathan, and we co-run a group together at MIT focused on molecular technology development for gene, cell therapy, and beyond. So our big thesis is how can we make biology as programmable as possible, and how can we make biology kind of affect every aspect of a person's life? So not just necessarily curing disease and improving health, but even beyond that into biomanufacturing, smarter materials, energy, climate, biodefense, and so on. And so, we use very basic protein engineering and sort of molecular technology development to accomplish those goals. And we've started out originally developing gene editing tools. So sort of as grad students during the original Cas9 CRISPR tech dev, and witnessing that whole revolution kind of take over the world, really, and trying to continue that trend of, if Cas9 can be programmable, what else can be? And so maybe I'll pass it to Jonathan to also introduce this.
No, I think that's a good summary of kind of what we're thinking about these days. It's obviously a very exciting time in the field with different technologies emerging, and we're starting to see, actually, from a translational standpoint, some maturation of earlier gene editing technologies. So it's becoming very clear what the current challenges are. And so now, as a field and as our group, it's really kind of on our shoulders to start to develop ways to address those challenges. So thinking about how we can manufacture things more easily, how we can do more complex edits to the genome in the case of genome editing therapies, how we can deliver things to cells in the body in a much more effective way. These are all things I think that are on top of everyone's mind as well as ours. So we're very excited to chat across these topics today.
Yeah, no, I think I definitely carry that enthusiasm about being excited about addressing new challenges which are in the space, and once certain challenges have been addressed, how can we take things even further and really take things to the next level? But before I guess we dive specifically into PACE, I'm just curious to ask, what eventually led you both to focus specifically on the gene editing side of research?
So happy to chime in on that. I think when I was coming up through sort of high school, college, I think there was just a lot of excitement about genome sequencing, right? Like when I was just starting high school, I think it was announced that we had completed the first draft of the human genome, and from there it was rapid sequencing of people across all sorts of diseases, and these things called genome-wide association studies, where you can sequence hundreds of thousands of people and say, hey, these mutations might be driving this disease, and these ones are driving this disease. And so there really was this race to start building gene therapies and ways to treat these diseases. And so for me, coming from an engineering background, both my parents are engineers, it was really interesting to think about this is the beginning of engineering cells and engineering DNA and kind of this new field. And so I was really excited to kind of figure out how can I contribute to this and engineer the unseen world, right? Like it's weird because you can't really see what you're doing. It's like reagents that are at the atomic scale, but you have to really believe that what you're engineering is there and have ways of reading it out so that you can know what you're actually doing. And that always fascinated me as well. And so when I was starting graduate school, I had just read this paper from Feng Zhang at MIT about this new bacterial protein called CRISPR-Cas9. And they had found a way to use it in human cells outside of their bacterial context and actually figure out a way to very easily reprogram the system to new genes to make cuts and try to correct genetic information. And it was fascinating that they had made it as simple as, so that anyone could do it in a matter of days. You can design an experiment and, try to edit your cells. And to me, that was fascinating because biology had always seemed so complex and they had found a way to make it less complex. And I wanted to learn how to do that and figure out what's the limitations. I read that paper. I was like, oh man, this is now solved, this thing I wanted to do. But then, I joined Feng's lab and you learn that it's really just the beginning. There's still 7,000 genetic diseases. We just started human trials the past couple of years for 2 diseases, right? CRISPR Therapeutics, I think, is doing sickle cell, and Intellia is doing transthyretin amyloidosis, and we have a long way to go to hit the many other thousands of diseases, and there's actually very nuanced limitations that we still have to address. So, pass it to John.
No, I think that covers it well.
Yeah, I find it a really interesting backstory. Being able to see that engineering background from your parents and essentially apply that same engineering mind frame and way of thinking to how can we, engineer things on a micro scale within ourselves, within our living beings. And I think it is quite fascinating because, as you mentioned, that there's so many genetic diseases, and even within, specific genetic diseases, even like say cysticfibrosis, for example, there could be multiple different things that can, reach that end stage disease. So being able to, engineer and edit our DNA to address some of these problems and conditions is quite a fascinating field, and I definitely echo that enthusiasm. And within that same lens of, engineering and gene editing, very fascinated to discuss this new paced, gene editing methodology, how it, I guess, compares and improves from previous methods such as like the prime editing methods. Could you please explain, like in simple terms for the audience, what paste is and essentially how it works?
So I think it's really nice to think about these technologies kind of in the context of the larger field of genome editing, right? And prior to really 2016, genome editing meant a specific thing, which is creation of a break in the genome that allowed us to change the genome through endogenous machinery. So using the cell's own repair machinery to repair that break in certain ways. So the idea is that if we can cause a break in the genome, and actually this is based on DNA repair pathway biology that far predates CRISPR and other genome editing technologies. But the idea is that we can create a break in the genome, and repair of that break can either create what we call an indel, which inactivates a gene, and that can be very useful therapeutically, or allow us to also introduce a repair template that then can be integrated into the genome, although in a somewhat inefficient process. So this is what really we wanted to do for many years, and the idea behind the power of CRISPR, at least back then, is that it's a programmable way by coding a set of 20 letters that match 20 letters in the genome to create a break. And this technology underlies many of the first genome editing companies, Editas, CRISPR Therapeutics, Intellia. Then in 2016, multiple groups actually had the innovation. Instead of editing the genome via creating a break, you could actually recruit things to the genome that allowed it to actually be edited via different mechanisms. So the first example of one of these concepts, base editing, was using the Cas9 or other CRISPR technology to take a enzyme to the genome called a deaminase, and that actually performed chemistry on the genome to change a single letter. And this is a really powerful approach. Obviously, it has a very intensely specific approach to changing just that specific letter without having to provide any sort of external material or template. This is also the technology that underlies much of the therapies being developed by Beam. But of course, you can imagine that it's limited. We can only change certain letters. In fact, these technologies, the first technology could only really do 2-letter changes, and the subsequent technology could only do an additional 2. But there's many different letters that we can't change. In addition, of course, you can't change, put in sequence or remove sequence. So then around 2019, David Liu at the Broad Institute at Harvard developed a technology called prime editing. And this was very ingenious. It takes the capability of being able to actually prime, this is where it takes the name, off of the genome and actually integrate a specific sequence into the genome of our choosing. And that can actually lead to an insertion, a base change, or a deletion. And this is a very flexible technology. It's the underlying basis of the Prime company, Prime Medicine. And it has certainly revolutionized the field once again. But the caveat there is that we cannot put in large pieces of DNA. And if you would like to be able to cure a disease where mutations occur in a certain gene, often with many diseases, there are many different mutations in that gene, and you don't want to have to develop a therapy for each individual mutation and fragment the patient population. So there's a thought, which is what's the next phase or kind of epoch in this genome editing? And it's the ability to take a place in the genome and insert into that place specifically a very large piece of sequence. So when we say large, we mean that in the sense of letters, and we're talking on the order of 100 to 1,000 or even 10,000 letters into the genome. And this allows us to put complete genes in, or in the case of other therapies like cell therapies, engineer these cells outside of the body in a very complex way, which allows us to have a large response. And that was really the motivation for us developing PASTE. And we'll touch on the technical aspects of PASTE later on, I'm sure, but just the overall concept of PASTE is that we want to programmably insert a large piece of sequence into the genome wherever we want. And so we can do that by combining different technologies for actually putting small things in the genome and then using those small things to integrate larger things in the genome. And this allows us to very efficiently edit the genome, both inside the body as well as outside of the body, for both gene and cell therapies.
Awesome. Science, it sounds very interesting. I like the fact that you both mentioned that these technologies have essentially existed within biology for such a long period of time. Conscious and aware that, with the whole CRISPR-Cas system, this is something which was essentially a defense system within bacteria, I believe, to protect them from viruses. But it is really interesting how, as you described, Jonathan, there's been this constant progression, both prior to 2016 and then after 2016, where we keep pushing that boundary of how we can be a bit more creative with the gene editing, further and further. Like you alluded to, being forced to edit specific letters or DNA base pairs specifically across a large gene might be a bit of a challenging task. But with technology such as PASTE, being able to insert now large pieces of genes, with very few amounts of underside deletions or insertions, I think that's going to be a very interesting technology, which is going to pave the way for interesting applications. And just going on from there, I was reading from one of your papers that essentially, with this programmable approach, and please correct me if I'm wrong, it essentially is combining this whole CRISPR-Cas system with a group of integrators, serine integrators from viruses. And I was really curious to ask, what were some of the challenges that were there in terms of taking this, serine integrase system from viruses, usually being applied against bacteria, and programming this to be used in human gene editing.
Yeah. So, what was the engineering process like? Or you mean just conceptually, how do we think about this stuff?
I think both. I think it's both interesting to hear about how the idea came up, and also the engineering side of things as well.
Sure. Well, I think the idea of gene integration has been on our mind for, probably almost 8 years now. And I think the reason is we realized early on that I mentioned there's 7,000 genetic diseases, but there's actually like millions of variants. Like if you look at ClinVar, which is the most comprehensive variant database, I think there's like 16 million unique entries now, and more than 20% are known to be pathogenic, which means there's a lot of mutations we have to correct. And if you have to go mutation by mutation, that's millions of drugs you'd actually have to build, which is just not sustainable. There's not enough resources for that. So we recognized early on that it would be nice to have enzymatic ways of, pasting new copies of genes into the genome. And we've explored a lot of those ways. One was trying to use ligases that actually ligate, you cut with Cas9 and ligate a new gene in, and that didn't work early on. And, after trying a lot of different things like transposases and recombinases, we landed on these serine integrases. And it's been long a challenge in this field to reprogram them, right? People have known some of these exist. There's YC31, BXB1. The literature goes back like maybe 15 years or something. And the problem is the protein its activity is so linked to this like 46 base pair site that it likes, that if you start tweaking the protein, you just start killing its activity. And so, we had tried to tinker with these for a while and we couldn't crack it. But when we saw the prime editing paper came out, we realized that things like Cas9 reverse transcriptase fusions, which allow you to copy in small sequences into the genome, could be paired with something like an integrase to basically make a new system that's capable of large integration. And the idea is if we fuse a Cas9 to reverse transcriptase into an integrase, the first two domains can copy in the small target site the integrase likes, and then the integrase can simultaneously integrate into that site the larger payload. And we actually started tinkering around with this concept, and we couldn't get it to work. We had used certain integrases called recombinases, like CRE, and we quickly realized that they're just not enzymatically suited for this process. People use CRE to recombine, sequences like invert sequences, but it's not good at combining two sequences together. But we're tinkerers, so we didn't give up. We started playing around with other enzymes, and we quickly realized that things like BXB1 do the job much better. And I think the first experiment we tried, we ended up getting like 10-15% efficiency. And from there, we were able to get it up to 40, 50, 60%. I think now we're even getting like 80% efficiency in cell lines. And so, I think it comes back to our engineering tinkerer mindset. We have sort of a high level concept, and we just start, exploring and kind of throwing the kitchen sink at it. And then hopefully we end up with something that works.
Yeah, no, I think it's a very exciting story, and I like the fact that at its core, it is that tinkering aspect of literally experimenting. Just thinking back some of the research that I've done in my past studies. Obviously, in no way compared to the work that you guys are doing. But there were always times in the lab where things would go wrong, or things were going well, but I wanted it to go better. And sometimes the result and the path to getting someone which was much better was simply by experimenting and using a fairly logical approach to try out different things. I really do like the thinking that you guys applied in terms of saying, if we're having this issue of not being able to make too many changes to this enzyme, in this case the serine integrase, and the site of the DNA that which it likes to attach to, why don't we just simply insert that site, that launch pad, into the area where we want to edit the gene?
Yeah.
And in hindsight, it might sound a bit for lack of a better word, simple, but I think it's absolutely genius. And it's one of the things where it solves a great problem, and it really advances research in that direction. It's really interesting to have about some of the other enzymes and recombinases that were tested out as well. And so just progress from there. I'm very curious. In both of your eyes, where do you see the strongest use cases for this type of gene editing technology within diseases?
Well, so, with insertion-based technologies, there's really, I think, two things that it catalyzes. One is, as Omar mentioned, the ability to edit diseases and cure diseases in a mutation-agnostic way. So when we identify a disease that's linked to a gene, instead of having to develop a therapy for a specific mutation, we can just integrate the gene itself in a very, kind of agnostic way. And this means that, one, we don't necessarily need to debug and produce a therapy for each site. And two, we can also overcome any challenges that are based on other variation between individuals. There's a large question on are other aspects of genetic variation between individuals other than the mutation that we're trying to treat itself. With CRISPR going to inhibit editing, for example, if there's a nearby site that, for example, a other base nearby that affects editing, we don't need to worry about any of that. So, for example, cystic fibrosis has 1,800 different mutations, and cystic fibrosis is an area that we're very interested in using PACE for. So there's a wide number of different diseases with this spectrum mutations, and I think that's kind of the first place that the mind goes when applying this therapeutically. At the same time, I think that the utility for genome insertion technologies and programmable genome insertion technologies in cell editing and development of very tailored and programmable cell therapies is also very enticing, where we can edit cells either outside of the body ex vivo or potentially inside of the body efficiently in vivo, and install bells and whistles that are useful for adoptive cell therapies or targeted cell therapies, such as chimeric antigen receptor, like for CAR T generation, for example, or other ways of modifying cells for immune or non-immune cell therapies. I think that the ability to edit the genome with this precision, with these large payloads, it will be very helpful as the actual cell editing approaches get more and more complex and have more and more components in them. So we're very excited on that aspect as well.
Yeah, no, exciting. It sounds like although it can be directly applied to treating some genetic diseases, that's essentially only the beginning. And like you said, it can be used in, I guess, helping the process of making CAR T cells and other applications as well. I'm very interested in how this type of technology can be used in diseases such as cystic fibrosis, which is quite a big issue, I guess, within the healthcare system, and also in diseases such as Huntington's disease. I've noticed that that was also mentioned within some of your papers. And just for the audience, for people who are listening or watching online, essentially Huntington's disease is caused by a mutated version of the Huntington gene, where essentially there are some DNA base pairs, specifically cytosine, adenine, and guanine, so CAG, and within a healthy gene, there's repeats with gene, let's say 10 to 35, but within the mutated version of this gene, there's essentially excessive repeats of this region, called a trinucleotide repeats, because of the 3 DNA base pairs. And essentially, when you have excessive numbers of repeats, above 35, people who have this mutated gene essentially develop Huntington's disease. And being autosomal dominant, it means that only one parent having the disorder means that there's a 50% chance of the child getting the disease. And I guess for my finance listeners, from the finance background, if you're curious to remember, there's specific DNA base pairs. You can imagine CAGR, remove the R, and there you have CAG. But just looking at scenarios like that, where, let's say, for example, the faulty gene has a region of DNA which is repeated, and even in the most extreme cases, if you have 100 of these excessive repeats, 3 DNA base pairs times 100 is approximately 300, and being able to go in and edit large sections of DNA would mean that potentially this technology could be used to solve this problem by editing or removing excessive or misbehaving, for lack of better words, genes within these individuals. So it is a really exciting field. And just following up on what you just mentioned about being agnostic, I'm really curious in terms of the areas of the genome that this technology can be used to target. Is there any kind of limitations in terms of the types of DNA regions they can target, or is it technology that can be used to target a very expansive range of DNA regions?
Yeah, I think that's a good question. I mean, so the way, any Cas9 or CRISPR-based technology works, right? There are some sequence specificities to these CRISPR enzymes. And what that means is there are some limits to where you can target in the genome. So, for example, Cas9 has to have a target site next to a PAM, which is a protospacer adjacent motif. And that PAM is of sequence NGG, which means you have to find 2 guanine nucleotides in the target region you want to target. Now, that doesn't always happen, especially if you're targeting a specific mutation. Like, your PAM has to be right next to that mutation, and there's sometimes a low chance of that happening or no chance. And so this is why over the past few years, there's been a huge push to find all sorts of new CRISPR variants, new types of Cas9 orthologs, even Cas9 engineering to try to change its path via protein engineering approaches. And that has worked to some extent. I think, with base editing technologies, they've drastically expanded the target space. But here's the advantage of doing what we're talking about, which is gene integration. Because we're replacing the whole gene, we have we don't have like a specific mutation we're targeting. We actually have a large part of the upstream region of the gene that we can target. In fact, the strategy we're targeting most of our gene replacements is by targeting the first intron of the gene. So the way we do it is we take the coding region of the gene, except the first exon. And so if you can imagine there's a gene sequence, right? You have the promoter, then you have the first exon, and then intron 1, and then the rest of the exons. And so what we're doing is we're targeting intron 1 with the cDNA of the protein, except for exon 1, and we're splicing into exon 1. And so we have the whole intron 1 region to target, and for most genes, that's like thousands of base pairs. We can target anywhere in there. So we actually, for programmable gene integration, there's so much flexibility that like the sequence constraints don't really matter compared to typical base editing and prime editing and all these other much more mutation targeted approaches, which is nice.
Yeah, that's very nice. It's very nice. And on that same point about the the increased flexibility and applicability of this paced technology and methodology, I'm really curious. I was reading that essentially you've recently had success in certain genes, success rating from 5 to I believe it was 60 or 65% success. I think you mentioned even in this talk that in cell lines you're commonly seeing success up to 80%. So I'm just really intrigued and curious to ask: are there certain types of genes or even certain types of cells where typically a greater success rate is seen?
Yeah, I think with any technology, genome editing, there are many factors that influence it. Of course, with technologies such as base, and also I'd say with prime editing as well, more broadly, cell types and organisms have both been shown to be very, very important considerations for efficiency. So, for example, in mouse cell lines, we see that we have reduced efficiency, and we also have more reduced fidelity of the landing site relative to human cells. So that's certainly a consideration. We have seen high efficiency integration across many different cell lines, but in certain cases, for example, primary hepatocytes, delivery is more of a challenge, and so therefore we get less of our template in, and therefore we have a less efficient delivery. But hepatocytes are one of the most relevant translational tissues, so we've been working very ardently to actually increase that rate to actually get that rate up via a lot of optimizations, especially on the delivery side. With locations, there are certain places in the genome that can be easier or harder. For example, cystic fibrosis, CFTR, the gene responsible for CF is notoriously actually difficult to edit with a variety of different technologies. And it took us a while to find a good targeting strategy, though we do now have one. So I think as this technology is, as with every genome editing technology, is adoption really increases, people will find these edge cases of the technology where things work well or don't work well, and that'll really add to the growing body of knowledge of how to optimize paste for different applications.
Exactly, and I think like you both alluded to earlier, in those scenarios and cases where certain types of cells or areas might be harder to target, whether that's the hepatocytes in the liver or whether that's for that CFTR gene for cystic fibrosis, it is an ongoing process, and it is just a case of further experimentation. And I'm very glad to hear that you found a way to target that cystic fibrosis gene. It's definitely definitely something very very nice to hear. Yeah, so I guess shifting things slightly to a slightly new tangent. As both of you are literally researchers working on the cutting edge of scientific technologies, what would you say, I guess, generally are some things within the field that excite you, whether it's within gene editing or even outside, that you're seeing in the research space?
Yeah, I think that's a great question, because I feel like we're constantly getting excited about new stuff, and probably changes depending on what month you ask us. But for this month, I would say probably what's happening in artificial intelligence and the intersection with biology. One, it's been fascinating, I guess, for the past couple quarters or maybe past year to see what AI has done for society, right? We went from, AI being kind of this fun toy, I guess, that people would play with online to now, like, it can predict or like make new types of art, right? Like there was all the generative and diffusion art that people were showing. Then there was like the language models like ChatGPT, where like you can talk to it and it talks back to you, which is crazy. And now we're starting to see that excitement kind of seep into the biology world, where over the past even few months, there's been diffusion models for protein generation, where you can be like, here's the shape of a protein I want, and it'll just like invent what the protein sequence should be, and it'll like fold correctly, right? Which is like kind of like magic in many ways. And it can be complex protein shapes like protein motors or protein like giant assemblies. Or we're seeing the ability, like large language models can be used for modeling all of proteins. Like Facebook has a model for this, or Meta, I guess is what they're called now. DeepMind has a model for this. Salesforce published a model a couple of weeks ago. And the things you can do with those models is also really cool. I think, even in our own lab, we've been adapting some of these models for rapidly evolving proteins. I think like CRISPR systems, some of the new ones we find, they're really tiny, like a couple hundred amino acids. And as a result, they're not that active. But using these AI models, we can actually rapidly evolve them by just having the model tell us, here's the mutations I think you should make. It's almost like you went to ChatGPT and you're like, hey, how do I make this protein more active? And it like would tell you some mutations. We go test them, and then we come back to it. And we're like, well, here are the mutations that work, and here's the ones that don't work. Can you tell us more to make? And it'll tell us more to make. And we do a few rounds of that. And now we have a protein that's like 1,000 times more active, which is, surprising us in the lab that even works. But so I think like stuff like that. There was a paper from Salesforce last week where they can hallucinate new protein sequences. So you're like, here's a family of enzymes and their sequences and their activity, and they'll make up new sequences that are like, more than 80% different in protein sequence, and they still like work, which is crazy. It's like, it's like almost like magic. I mean, maybe it's not magic. It tells you that probably like the way the protein space is, there's a lot of flexibility. There's a lot of ways to make the same protein, but it's cool that we can learn the language of protein by studying like hundreds of millions of proteins. And by studying, I mean making a neural network, aka a transformer model that models that. We have no idea how those work or what exactly it's modeling, but so there's some information in there that can basically tell you, here's a bunch of other ways to make a protein that does a desired function. So like, I think AI is even teaching us a little bit about protein evolution and evolution in itself, which I think is fascinating. So, it's a rapidly evolving space. It's super exciting to kind of be a part of it and to see where it'll go.
Yeah, that's very remarkable. I'm really intrigued. So in the example you mentioned about I believe you said it was like protein hallucinations. Is it essentially going to this AI model and asking, I want a protein that does this, or like I want a protein which is an enzyme which cuts this, and it pretty much creates the whole sequence, the whole folding structure and everything is?
Yeah, it can tell you the sequence, and then, I mean, the large language models don't necessarily tell you the structure, but once you have the new protein sequence, then there are things like AlphaFold, right? And these different models actually trained in similar ways that can then tell you the structure, right? But this is the crazy thing. Like, these models don't even need to know the structure of the protein. They've just learned like the sequence. It's like I gave you the entire grammar and all the words in the human language, and then this model could tell you new words that actually have useful like meanings and functions to fill in gaps in our language, kind of. I don't know if that's a great analogy, but I'm trying to explain it. But basically, right, it's just learning like what amino acids should go with each other, and it can then make functional new proteins given a set of functional proteins. Yeah.
Yeah, no, it's awesome, and I think it's really exciting to see that, you know, as time continues, we'll see more and more of AI being not just, you know, spoken as a buzzword, but actually being used with tangible examples. Similar to what you mentioned before about the human genome and GUS studies. The more time continues, the more these things become more relatable, more accessible, and it becomes a situation where it becomes more tangible in day-to-day life. And being able, like you said, to use an AI tool to help supplement research and to provide ideas which, quite frankly, would be hard to create from the normal human thinking process because it's coming from an alternative ideation and thinking process. I think is a wonderful thing, and it's only going to lead to the betterment of research, and as a result, the betterment of products, therapies, and improvements within healthcare. So it's really, yeah, it is a really interesting thing to see, and I think AI in life sciences and healthcare is a very interesting technology and trend indeed. But within that same kind of lens, again, as researchers, what would you say are some of the is one challenge that you see within the space? I know you've mentioned that within some of these gene editing technologies, certain genes could be a bit hard to access, or certain cell types, for example, with hepatocytes, could be a bit hard to access. Are there any kind of challenges that could kind of stand out and appear to be kind of the mother of all challenges within this space?
Yeah, I mean, that's a fantastic question, and I think the challenge that is at the front of many people's minds is delivery. If you look at either things that are in the clinic now or in people's preclinical pipelines for gene editing, they seem to follow a lot of the same trend, which is they target the liver or the eye or ex vivo. And that is not a coincidence. It's based on the filtering that if we're going to be putting something into a patient, we need to be able to get it into their cells in a effective and safe way. So I think that delivery technologies, obviously in the past 2 to 3 years, we've seen a huge renaissance, a practical renaissance in lipid nanoparticle technologies, where the Pfizer, BioNTech, and Moderna vaccines are far and ahead, the largest application of these approaches. But now there's a large amount of capacity to manufacture LNPs, and people are becoming more interested in how they can be applied. But getting LNPs to sites other than the liver, for example, or taking some of these kind of more early stage approaches to targeting brain or muscle, where there are certainly preclinical applications of things like AAVs, adeno-associated viral vectors, but really getting those to the translational part, maturity will be challenging. And there's many companies in the space. You see many coming out every day, but that, I think, is the next, really going to be the battlefield. And I would say that's half technical. How do we get them there? But also it really features into this larger conversation of cost and price of these therapies. It obviously is very difficult to price a one-time therapy that's curative. But a lot of that can come down to also the cost is very high. Then actually manufacturing viral therapies can be very expensive. So new approaches for biomanufacturing that reduce the cost of manufacturing these therapies will be able to make them more accessible, or we can actually make much more equitable to have gene editing or cell therapies in many different markets. So I think these go hand in hand and really are going to be some of the largest challenges the field will face in the next decade.
Yeah. It's I think the fact that you mentioned delivery is very interesting because there's a big challenge within this whole gene editing space. It's also in a sort of way can be a challenge within even like cancer as well, where you could potentially have a therapy which is very good at killing things and killing cells, and therefore would be good for kill cancer cells. But it's a question of how can we deliver it specifically to the right place and not have collateral damage, which is, I guess, a slightly different lens because it's more about how to have that targeted delivery, but it's still the lens of how can we deliver things specifically to certain cells. And in the discussion we're having today about cells which are hard to access, like liver cells and then the eye, for example, it is an interesting field where being able to address that challenge, I can imagine, would have a massive impact. But yeah, no, I think I think on a similar-ish lens, just thinking about this technology as a whole and the fact that it's used to insert whole genes, do you have you had any thoughts or ideas or any concerns about the potential of this technology being used for more like cosmetic means? So, for example, trying to change certain attribute like eye color, for example, or even like genes related to like athleticism, such as myostatin related genes as well. Or was it more of like a sci-fi fiction thing, which doesn't really come to mind?
Yeah, I mean, I think if we can understand, positive attributes, non-therapeutic uses of genetic modulation, I think that would be beneficial for society. I mean, we already take vitamins and supplements and, things that can improve our health. And I think, the permanence of gene editing makes some of those things scary because we may never fully understand the impact of like a mutation that improves muscle mass. Like maybe that's not something you want permanently in your body. This is part of the reason why we also develop a lot of RNA editing technologies, right? So our group and our work has not just focused on DNA, but like how do you actually make mutations and insertions, deletions in RNA? And RNA is great because if you withdraw the technology making the edit, it'll revert back to normal after a day or two. So we're very interested in this way of doing reversible gene editing, so to say. And that can not just be useful for like things like improving bone mass and muscle and cognition, but also things like modulating the immune system for infectious disease or modulating pain or regeneration, like after a heart attack or a liver injury. And so, yeah, we think about that problem and we're thinking about ways to actually, make it potentially practical one day.
Yeah. I think on the cosmetic front also, it's interesting to imagine the possibilities, but just from a, perspective of feasibility, not necessarily technical, but economic, I'm not sure how many people would spend, on the excess of $1 million and probably with some sort of potential adverse side effects to just have their eye color change. So, which is why I think even with other diseases beyond, cancer obviously is a disease that is immense burden to the patient and, deadly. So that's one that I think has a lot of promise because, safety is a consideration, but there's much more beneficial use there. But as you even go to other things like autoimmune, it becomes more of a balancing act. Where does the patient want to be able to take on that risk to themselves? So I think it's always important to think through, when you go and have one of these therapies, how much is it going to cost, or what's the price going to be to the patient? And also, what are the potential adverse risks that these patients will want to incorporate into whether they actually elect to take the therapy or not?
Yeah, no, I think it is a really good point, really good and important point. And even echoing from that, on that specific point about the cost of therapies, reminds me of what you was mentioning just a moment ago. is really interesting because one of the researchers at MIT, I believe his name is Andrew Lowe, he has some very interesting research about how potential financial systems could be used to fund certain, research for disease areas where, independently it would be too expensive to do the research, but basically having them combined in a more of a combined system where essentially only a few of them need to have successful outcomes in the research to enable the whole part of the research to be economically viable. It's quite an interesting way to address some of these challenges concerning, therapy areas and research for diseases, which are hard to fund, essentially. But anyways, conscious of time, so I'm going to bring things to a close. But thank you so much for your discussions today, Omar Jonathan. Really appreciate the call, and I'm interested to keep in touch in terms of the research and the new additions in this space. I'm going to turn to the audience today, but before I do that, if people want to reach out to yourselves, what is the best way to get in contact?
Probably our emails, which you'd be happy, I don't know, to share. You maybe fan to post, or and our emails are also readily available on our website as well.
Awesome, thanks. Wonderful. Okay, so thanks again for joining the podcast. It's been a fun episode, and for everyone who's either listening or watching online, please do like, share, subscribe, so more people can have access and exposure to these interesting technologies and emerging trends. I look forward to speaking to all of you soon in the next episode, and thank you.